The shroud was not a painting, not a scorch, or not a photograph.
Well, you see this when you see the shroud in negative. Like, Jamie, pull up an image of it. It's very strange. Like, it shows the lash marks on his body.
Yeah.
It shows his facial features. It shows the holes where his wrist is, where he was crucified.
What if the world's most controversial religious artifact just revealed a hidden biological secret that science can no longer ignore? For centuries, the Turin Shroud has divided humanity. Some call it the burial cloth of Jesus Christ. Others dismiss it as an elaborate medieval fake. But now, a shocking DNA discovery buried deep inside its ancient fibers is forcing scientists to confront questions they never expected to ask. Before we expose the discovery that left researchers stunned, hit like and subscribe, because some believe the truth behind the shroud is something powerful institutions never wanted fully revealed. This is not just another ancient relic story. This is where faith collides head-on with modern science. Using advanced genetic analysis, forensic imaging, spectroscopy, and high-energy testing, researchers uncovered microscopic traces locked inside the cloth itself. What they found was far more complex than anyone imagined. DNA signatures from multiple regions of the world, mysterious biological markers, and patterns that scientists still struggle to fully explain. For believers, this could be the closest physical connection ever found to the final moments of Jesus Christ. For skeptics, it raises even bigger questions about how such an object could have been created centuries ago, with details modern technology can barely reproduce. For over 600 years, the Shroud of Turin has remained one of history's greatest mysteries. But now, science may have uncovered evidence that changes the conversation forever. Researchers stopped seeing the shroud as a sacred icon. They examined it as a crime scene, as evidence in an unsolved case, as a biological hard drive, silently recording 2,000 years of history. They extracted and sequenced the DNA embedded in the fibers. They scanned every single thread with X-rays. They broke the bloodstains down into molecules, atoms, and elemental signatures. They expected simple answers, traces of a single individual or perhaps remnants of pigment left by a medieval artist. But what they found was far from simple. The genetic analyzers at the University of Padua and the physicists working in the sealed laboratories of ENEA discovered results that left even the most experienced scientists shaken. The data didn't just question existing theories, it shattered them completely. Both skeptical explanations and traditional religious interpretations collapsed under the weight of the results. The shroud was no longer just fabric. It became a map tracing a journey that began 2,000 years ago, a record of suffering so precise it cannot be reproduced, and evidence of an intense burst of energy, one whose nature modern physics still cannot explain. This marks the beginning of the most extensive forensic investigation ever undertaken. We will follow trails invisible to the human eye, microscopic pollen grains, molecular residues, and biological fingerprints. Step by step, we will uncover whose blood truly soaked these threads and why it seems to scream of torture, spoken not in words, but in the language of biochemistry. We will examine the physics behind the image itself, an image that functions as a photographic negative, a hologram, and an X-ray all at once. We will also answer a question that stunned the scientific world. Why DNA analysis forced researchers to admit that this artifact could not have been created in Europe, and that it bears witness to events that altered human history. To truly understand the shock of modern genetic findings, we must return to the very beginning of scientific shroud research, modern syndenology, back to the moment when humanity first truly saw the face hidden within the shadows of the linen. That moment came on May 28, 1898, in Turin. A lawyer, city council member, and passionate amateur photographer named Secondo Pia was granted rare permission by King Alberto I to photograph the relic during its public exhibition, held in honor of the prince's wedding. At the time, photography was far from simple. It was slow, technical, and demanding. Pia hauled a camera the size of a suitcase onto tall scaffolding inside the cathedral. He relied on powerful magnesium flashes and lamps to cut through the dim interior. He exposed 2 large glass plates, each measuring 50 by 60 centimeters. Late that night, alone in his home darkroom, illuminated only by the faint glow of a red lantern, Pia lowered one of the plates into a bath of developing chemicals. And then it happened. As the image began to emerge on the glass, Pia nearly dropped the priceless negative in shock. What he saw felt unreal, almost supernatural. On the photographic negative, where light should turn dark and dark should turn light, there appeared not a faint, blurry stain like the one visible on the cloth itself, but a sharp, high-contrast, astonishingly detailed positive image. A face, deep-set, closed eyes, a broken nose, a mustache, and forked beard, bruising on the right cheek. For the first time, the figure hidden in the linen was no longer a shadow. It was a man. The face carried an expression that was majestic, calm, and commanding. Yet it belonged to a man who had just endured unimaginable physical suffering. The impact was overwhelming. It wasn't just a surprise. It was a revolution in how people understood the shroud. A normal drawing, whether made with paint, charcoal, or even blood, behaves predictably when turned into a photographic negative. Light areas become dark. Dark areas become light. The result is distorted and unnatural. The face turns into a flat, mask-like image. But the shroud was different. When photographed in negative, it looked like a true, black-and-white photograph of a real, living person. That single fact changed everything. And to this day, it remains impossible to dismiss, no matter how skeptical or determined to debunk it someone may be. What we see on the cloth with the naked eye is not a normal image at all. It is already a negative. And that raises an impossible question. Who in the Middle Ages, in the 10th or 11th century, understood the principles of photography? Who knew that light could be reversed to create a hidden image? The answer is simple. No one. Who could deliberately create a perfect negative image without any way to preview or verify the result? Photography would not exist for another 800 years. The human eye and brain are not biologically capable of perceiving the world in negative, let alone reproducing it with such precise gradations of light and shadow. This was the first true mystery, the first crack in the seemingly solid wall of skepticism. The shroud did not behave like a painting. It behaved like a photographic plate, capturing a single moment in time. Over the years, the cloth has been examined with X-rays, ultraviolet light, infrared imaging, and laser scanning. Yet the greatest secret was never in the image itself. It was in the dirt, in the dust, in the microscopic particles trapped between the linen fibers during 2,000 years of movement, handling, and exposure. In 2015, a team of geneticists and biologists led by Professor Gianni Barcaccia of the University of Padua was granted unprecedented access to the relic. Their mission was as bold as it was unsettling: to locate and analyze DNA. They were not searching for the DNA of God. Science doesn't know what such a trace should look like. There is no reference sample, no standard to compare it against. The researchers weren't trying to identify a single individual. They wanted to reconstruct the history of the cloth itself, where it had been, who had touched it, and whose hands had held it across centuries. To do this, they used specially designed, sterile micro-vacuum devices fitted with ultrafine filters. These tools gently collected microscopic particles of dust, pollen, and organic material not only from the surface of the fabric, but from the deep spaces between the warp and weft threads, where ancient traces can remain sealed for millennia. They also analyzed particles gathered during the 1988 restoration, which had been carefully preserved in archival storage. This work pushed the limits of what science could safely attempt. Even the smallest modern contamination, a sneeze, a breath, a single flake of skin from a priest, conservator, or technician could erase ancient signals and replace them with modern genetic noise. The samples were transported to an ultra-clean, Grade-A laboratory. There, the sequencing began using next-generation sequencing, or NGS. The scientists focused on mitochondrial DNA from both plants and humans, and for a crucial reason. Unlike nuclear DNA, mitochondrial DNA exists in hundreds of copies per cell, is passed down exclusively through the maternal line, and survives far longer in ancient material. It serves as a powerful and reliable marker of geographic origin and human migration. For weeks, computers ran non-stop, decoding millions of nucleotide sequences, comparing them against global genomic databases representing populations from across the world. When the final diagrams appeared on the screen, maps of genetic haplogroups and ancestral lineages, the researchers realized they were looking at something that defied any simple explanation or forgery. This was not the genetic profile of one person. It was a genetic portrait of humanity itself. The findings were published in the highly respected journal Nature Scientific Reports, and the reaction was explosive. The results sent shockwaves through the scientific community. The scientists expected to find one clear, dominant genetic signature. If the shroud were a medieval forgery created in France, as skeptics have long argued, then European DNA, specifically French or Italian, should have overwhelmingly dominated the results. If, on the other hand, it were an authentic relic from Jerusalem that had never left the region, then the genetic traces should have pointed almost exclusively to the Middle East. That is not what they saw. What appeared instead was chaos. The entire world written onto a single piece of cloth. The shroud revealed genetic fingerprints from people across vast regions of Eurasia and Africa. Let us examine what they found. First, the Middle East. Scientists identified haplogroups commonly associated with the Druze, a tightly knit, historically isolated ethno-religious community living in the mountainous regions of Israel, Jordan, Lebanon, and Syria. Druze DNA is considered extremely ancient and remarkably stable, having changed little over thousands of years due to centuries of isolation. This makes it a powerful marker of geographic origin and provides strong evidence of a Middle Eastern connection. Second, Western Europe. Haplogroups such as U5b and H1 to H3 appeared exactly as expected. Since the 15th century, the shroud has been kept in Europe, first in Chambéry, then in Turin. It was handled by the Poor Clare nuns who repaired it, by members of the House of Savoy who owned it, and by countless European pilgrims over the centuries. Third, North and East Africa. Haplogroup L3CM1A1 emerged, pointing to regions such as Egypt and Ethiopia. This was unexpected and deeply intriguing, suggesting contact with some of the world's earliest Christian communities in Africa. Fourth, South Asia. Haplogroups M39, M56, and R8 appeared, genetic markers typical of the Indian subcontinent. And most astonishing of all were traces from East Asia. Haplogroups such as D4 and G2A, markers associated with China, were present. China, India, Africa, Europe, the Middle East, all of it encoded into one ancient cloth. How could this be possible? If the shroud were a forgery created in the damp basement of a French abbey around the year 1350, how could traces of Chinese and Indian DNA possibly be present on it? In the medieval world, globalization did not exist. There were long-distance contacts, yes, but not on a scale vast enough to leave such widespread, clearly identifiable genetic traces on a single piece of cloth. The answer turned out to be even more remarkable than the mystery itself. The shroud is not merely a burial cloth, it is a traveler. When scientists in Padua mapped the genetic data, they made a stunning discovery. The genetic distribution aligned almost perfectly, with precise geographic accuracy, with an ancient historical route that many scholars had long dismissed as legend, the journey of the man-made image known as the Mandelion. According to early Byzantine, Syrian, and Arabic sources, the shroud was folded in four so that only the face was visible, displayed within a special frame. This folded relic, called the tetradiplon, did not remain in Europe. It moved across the ancient world. The journey began in Jerusalem, the place of death and resurrection. From there, it traveled to Edessa in the 2nd century. The shroud remained there for centuries, hidden within the city walls, before being rediscovered. Edessa stood at a vital crossroads of the ancient world. The Great Silk Road. Caravans from China, India, Persia, and Arabia passed through the city. Merchants carrying silk and spices, pilgrims and diplomats all came to venerate the relic believed to protect the city. They breathed near it. They kissed its casing. They touched the reliquary. And with each encounter, microscopic traces, skin cells, hair fragments, and sweat settled onto the cloth. Layer upon layer, century after century, the DNA of the world accumulated on its surface like invisible dust. The journey continued. From 944 to 1204, the relic was held in Constantinople, the heart of the Byzantine Empire. The emperors of Byzantium acquired it by purchase or by force and carried it to what was then the capital of the world. Constantinople was a true mega-city of the ancient world, a place where peoples of every race, culture, and region came together. Then, in 1204, during the chaos of the Fourth Crusade, the city was violently looted. In the aftermath, the shroud vanished. Around 1205, after the fall of Constantinople, the relic passed into the hands of French knights and was kept in Athens for a period of time. Then, in France, around 1353, came the shroud's first clearly documented appearance in Western Europe, in the possession of the knight Geoffroy de Charny. And this is where the genetic evidence becomes devastating to the medieval forgery theory. A forger working in 15th century Europe could not possibly have gathered dust and DNA from people in China, India, the Middle East, and Africa, regions he knew only through distant travelers' tales like those of Marco Polo. Nor could he have deliberately planted those traces onto a cloth in a way that would deceive genetic scientists 600 years later. The DNA found on the shroud is not random contamination. It is the collective biological memory of humanity. It is dust from the sandals, robes, and hands of countless people who gazed upon this face over thousands of years. This biological record proves the antiquity of the object and its Eastern origin more convincingly than any radiocarbon test ever could. But human DNA is only half the story. The other half is written in plants. Palynology, the study of pollen, offered another crucial piece of the puzzle. Two respected experts approached the mystery independently. Professor Avinoam Danin, a renowned Israeli botanist from the Hebrew University of Jerusalem, and the Swiss forensic scientist Max Frey, one of the pioneers of forensic pollen analysis. Working separately, they examined pollen grains trapped deep within the linen's fibers. Using adhesive tapes, they carefully lifted microscopic samples from the fabric. What they discovered caused a sensation in the botanical community. Pollen from 58 different plant species was identified on the shroud. 17 of those species are native to Europe, exactly what one would expect, given the shroud's documented history over the past 6 centuries. But the majority came from elsewhere. Plants native to the Middle East, Turkey, and the Anatolian steppe, perfectly matching the ancient route through Edessa and Constantinople. Then came the most striking discovery, a group of plants that grow nowhere else on Earth. Species found only in a narrow geographic corridor between Jerusalem and Jericho. At the center of this discovery was one plant in particular, Gundelia tournefortii, a thorny desert shrub, a type of thistle. Its pollen accounted for nearly half of all the samples found on the cloth, an enormous and highly unusual concentration. Why would pollen from a thorny plant be so dominant? Why would thorns be associated with the burial cloth of a man treated as noble? The answer emerged when historians and theologians revisited the descriptions of Christ's Passion. The crown of thorns. Gundelia tournefortii, with its long, rigid, needle-like thorns, is precisely the kind of plant Roman soldiers could have used to twist together a mocking crown for the King of the Jews. And suddenly, the pollen began to speak. This plant blooms around Jerusalem at a very specific time, early spring, during the Jewish festival of Passover, Pesach. The discovery of an unusually large concentration of pollen from this exact thistle, concentrated around the head and shoulder areas of the cloth, serves as direct evidence that the body was crowned with it. A second botanical clue came from Zygophyllum dumosum. This plant is endemic to a very limited region, growing only in the Judean desert and parts of the Sinai Peninsula. Its pollen was also found on the fabric in significant amounts. No medieval forger working in France could have obtained pollen from plants native only to Israel and then applied it invisibly at a microscopic level onto a piece of cloth. A forger would have used paint. Pollen cannot be painted. It acts like an invisible seal from the crime scene, a form of geographic fingerprint that cannot be forged. For many years, especially during the highly rational 19th century, skeptics argued that the reddish marks on the shroud were nothing more than pigments, ochre, cinnabar, or tempera mixed with gelatin. That claim collapsed in 2017. A team of Italian researchers, led by Professor Giulio Fanti of the University of Padua, working alongside physicians from a hospital in Trieste, examined the stains using transmission electron microscopy and Raman spectroscopy. By analyzing the material at the nanoscale, they saw not pigment, but blood. Human blood, group AB. It is one of the rarest blood types, yet it appears frequently on ancient Christian relics, including the Sudarium of Oviedo. But this was not the blood of a healthy person. Within it, scientists identified nanoparticles of creatinine and ferritin bound to hemoglobin. Such extreme concentrations appear only under one condition: severe, fatal trauma. Prolonged torture accompanied by dehydration and massive muscle damage. When muscle tissue is destroyed by repeated blows, a process known as rhabdomyolysis, Creatinine floods the bloodstream in enormous quantities, and the blood on the cloth recorded exactly that. This is a biochemical scream of pain. The analysis revealed that the man wrapped in this cloth did not simply die. He was beaten to a condition that was already incompatible with life before the crucifixion even began. This aligns perfectly with the Gospel accounts of the Roman scourging, carried out with flagella, leather whips embedded with lead weights. The body shows evidence of more than 100 blows. The chemistry of the blood tells a story of suffering that no paintbrush could ever reproduce. An artist can paint the appearance of a wound, but no artist can imitate the biochemical signature of a body in extreme trauma, polytrauma, kidney failure, and hypovolemic shock. Another long-standing mystery was the color of the blood. The stains on the cloth appear red. Normally, ancient blood turns dark brown or black over time, but scientific analysis revealed unusually high levels of bilirubin, a substance released by the liver during extreme stress and severe trauma. Bilirubin preserves the red coloration of blood for centuries. The blood of a tortured man remains red. This is not mysticism. It is biochemistry under extreme stress. At this point, many ask the same question. What about the famous radiocarbon dating? In 1988, radiocarbon testing appeared to settle the debate once and for all. 3 of the world's leading laboratories, Oxford, Zurich, and Arizona, announced their results with 95% confidence. According to their measurements, the fabric dated to between 1260 and 1390, the Middle Ages, a forgery. The world accepted the verdict. Even the church stepped back, referring to the shroud as an icon rather than a relic. But science never stops evolving. 30 years later, researchers identified where the fatal mistake had been made. The error was not in the technology, it was in the human choice of sample. In 1988, a tiny piece of cloth, no larger than a postage stamp, was cut from the very edge of the shroud. That corner had been handled countless times over the centuries, grasped by bishops and cardinals during public displays. Sweat, skin oils, candle wax, and bacteria had all accumulated there. That edge suffered more contamination and wear than any other part of the cloth. Further research by chemist Ray Rogers of Los Alamos revealed something even more damaging. That corner had been expertly repaired during the Middle Ages, so skillfully that the repair went unnoticed. The sample was not representative of the shroud as a whole, and with that realization, the certainty of 1988 began to unravel. The monks who repaired the Turin shroud centuries ago did not use modern, invisible mending techniques. Instead, they strengthened the burned and damaged sections by carefully weaving newer cotton fibers into the original fabric. To hide the restoration, the cotton threads were dyed to resemble the aged linen, while gum arabic was used to bind everything together seamlessly. Years later, when scientists re-examined the exact sample used in the famous 1988 carbon dating test, they discovered something shocking. Cotton fibers were present in the tested material. That immediately raised concerns because the main body of the Turin shroud contains only linen, not cotton. In other words, the sample chosen for testing may have come from a repaired medieval section rather than the original cloth itself. That changes everything. If the laboratories tested a repaired patch mixed with newer material, then the dating result reflected the age of the repair, not necessarily the age of the shroud. Critics compared it to trying to determine the age of an ancient statue by analyzing modern glue stuck to its surface. What was once considered definitive proof against the shroud suddenly looked far less certain. Because of this controversy, researchers searched for a completely different way to study the cloth, one that could bypass contamination altogether. In 2022, Italian physicist Liberato De Caro introduced a revolutionary method known as wide-angle X-ray scattering, or WAXS. Instead of testing surface contamination, this technique examined the linen fibers themselves at the atomic level. Over centuries, cellulose inside linen naturally breaks down. Heat, humidity, radiation, and time slowly alter the molecular structure of the fabric, acting like a hidden clock embedded within the material itself. De Caro compared the shroud's fibers to ancient textiles with verified historical dates, including linen used to wrap Egyptian mummies and fabrics recovered from archaeological sites across the Middle East. The results stunned many researchers. The molecular aging of the Turin shroud did not resemble medieval cloth at all. Instead, its structure closely matched linen fragments discovered at Masada in Israel, textiles dated between 50 and 74 AD, the exact period associated with the life of Jesus Christ. Suddenly, the story changed. In 1988, many believed science had closed the case against the shroud. But decades later, advanced technology reopened the mystery. Yet the greatest question still remains unanswered. How was the image created? The figure on the shroud is unlike any known painting. There are no visible pigments, no brushstrokes, and no ink embedded in the fibers. The image exists only on the extreme surface of the linen, about 200 nanometers deep, far thinner than a strand of human hair. Remove the outermost fibers, and the image disappears completely. Scientists discovered the discoloration came from oxidation and dehydration within the fibers themselves, almost like a scorch mark produced by an unknown burst of energy. Researchers attempted to recreate it using heat, chemicals, radiation, and lasers, but nothing fully matched the mysterious image except extremely short pulses of intense ultraviolet radiation, technology far beyond anything available in ancient times. To recreate the full-body image across the entire cloth, experts calculated that an enormous release of energy would have been required, occurring in less than a billionth of a second without burning the fabric itself. Then came another astonishing discovery. In 1976, image analysis technology originally developed for NASA revealed that the Turin shroud contains accurate three-dimensional information. The brightness and depth of the image correspond precisely to the distance between the body and the cloth. No known medieval artwork possesses this property, and even modern artists struggle to reproduce it. Researchers also reported traces of Middle Eastern pollen, dust consistent with Jerusalem limestone, and blood evidence linked to severe physical trauma. Even the wounds appear anatomically precise. The nails are positioned through the wrists rather than the palms, matching modern forensic understanding of Roman crucifixion methods. Piece by piece, multiple scientific disciplines, chemistry, physics, biology, archaeology, geology, and history, continue pointing back toward Jerusalem during the 1st century. The Turin Shroud remains one of the most debated artifacts on Earth. Some see it as proof of a miracle. Others believe an explanation still waits to be discovered. But after centuries of investigation, one thing is certain. The mystery is far from over. So the question remains, will science eventually explain what happened to the Turin Shroud? Or are some mysteries destined to remain beyond human understanding?